In previous videos, we talked about complement activation, how there are three different pathways by which complement could be activated on a pathogen. Again, not every active pathogen activates every pathway of complement. So there's a lot of redundancy in the body depending on which pathogen can be recognized. So there's the alternative pathway of complement activation where the C3 protein spontaneously converts to IC3 and initiates the alternative pathway, alternative C3 convertase formation. There is the lectin pathway by which mannose binding lectin is produced by the liver and binds mannose on the surface of pathogens. There's the classical pathway of complement activation by which we spoke about the protein CRP, C-reactive protein, that is produced again by the liver in response to infections, and how CRP can bind phosphorylcholine on the surface of pathogens. Those are three mechanisms by which a complement can recognize a pathogen. We're going to talk about one more mechanism in the classical pathway. That allows for the body to recognize a pathogen. So, and again, it goes back to, since we're talking about the classical pathway, using CRP, right? C-reactive protein, again, which is produced by the liver upon infection, where when macrophages detect infection using IL-6, or using toll-like receptors, for example, macrophages release IL-6, which is a cytokine which can go to the liver and stimulate the liver to produce CRP, C-reactive protein, in response to an infection. And we talked about CRP interacting with phosphorylcholine, and we talked about this in a previous video, and this will attract the complement protein complex C1. C1 made of a number of proteins, C1q, C1r, and C1s. And in a previous video, we spoke about how that can lead to the formation of the classical C3 convertase. Now we're going to talk about how antibodies can be used to activate complement. So antibodies are produced in response to a pathogen via humoral immunity. So requiring B cells to recognize a pathogen's presence, have a immunoglobulin on the surface of that B cell with an antigen receptor, antigen binding site that can recognize an epitope on the surface of that pathogen. We'll cover that in great detail in later videos of humoral immunity. But if the B cells in the body are able to make antibodies that recognize a pathogen, coating a pathogen with antibodies, specifically IgM and IgG subtypes, that can attract complement a um, to a pathogen. So let's see that now. So the antibody we're going to talk about first, subtype IgM. IgM is one of the first antibodies. It's the first antibody made and released by B cells in response to infection. So when it's made, it forms pentamers. So there are five antibody molecules in a complex. And that's what I've drawn a pentamer there. You can see the 10 antigen binding sites, so the little ends of the Y's there. IgM is typically found in the bloodstream. It's a very large complex, so it is unlikely to diffuse into tissues, but it can be used to recognize pathogens in the bloodstream. So if there's a pathogen in the body and B cells recognize that pathogen and produce IgM protein, IgM antibodies, those IgMs could bind the surface of pathogens. Now, how is that going to help complement? Well, the C1 molecule can also bind IgM. So it can bind CRP and it can bind IgM, specifically the Fc region of IgM. And we'll talk about regions of antibodies in later videos. But for now, understand that the C1 complex, and again, it's those C1q stalks that sort of stick down, will interact with the Fc region of IgM antibodies, and that is going to do the same thing that C1 did when it bound to CRP on the surface of a pathogen. It's going to activate the proteases C1R and C1s, which will cleave C2 and cleave C4, and you have the production of the classical C3 convertase on the surface of a pathogen. We've covered that in previous videos. So antibodies can be used to recognize a pathogen, and complement will come and activate on that pathogen. So that occurs when IgM is produced. It can also occur when IgG is produced. So IgG is another antibody isotype, which is produced later upon infection. It requires isotype switching and affinity maturation to occur first. We'll get to those in later videos. IgG acts as a monomer. It's a small antibody that can diffuse into tissues, into lymph tissue, lymphatic system, lymph fluid. So you can find this antibody in a lot of places. And again, if the body has responded by detecting a pathogen and making antibodies, now IgG takes, could take quite a while, a number of days, a week, 2 weeks to make significant level of IgG protein. So again, this could take place quite a bit later upon pathogen exposure. But if the body has eventually made IgG antibodies, which work very nicely, they have very high affinity typically to the pathogen, and this antibody can get into lots of different places because it's small. Um, IgG will bind to the surface of a pathogen, and again, the C1 molecule can bind to the FC regions of the IgG proteins. And when C1 binds, we again know what happens. The C1R, C1S proteases activate, cleave C2, cleave C4, and again, you have the formation of the classical C3 convertase. So these three things, CRP, IgM, and IgG, all of them can attract C1 complex and initiate the classical pathway of complement activation. So that covers the different recognition mechanisms of complement.